So now we're starting the recording. So we started the urinary system. Again, renal anatomy, basic anatomy, nephron physiology, which is the microscopic component, as well as what is urine, what's in urine, and what are some of the things we evaluate with urine. So we'll go part one first. So on here.
Your volume is cutting out.
Oh, thank you. Thank you for letting me know. Let me see, hold on. Okay, let me try this again. I forgot when I do a share, there's a little clip button I have to push. Okay, now I've pushed the button. Now, hopefully this streamlines the audio a little bit better. Thank you for letting me know that. So let me know if the audio gets kind of wonky again. So as far as renal anatomy goes, you should be familiar with all of the terms as far as cortex and the medulla. What's in the medulla, what's in the cortex. The cortex, there's nothing else as far as gross anatomy goes, but you should know the nephron anatomy. Oh, okay. Oh, the video is still bad, or the audio is still bad.
It's actually the video that was cutting out.
Oh. Okay. Okay, thank you. The audio is good, but the video, okay, that's weird. Okay, so I'm just going to leave it on this slide. So you should have this in your notes as far as a checklist for part one. And if anyone wants to go, and I'll just kind of go through each line item. And if anyone wants to go through it in more detail, then we could move forward and see some more images. So that way you're not really, if the video's being wonky, you're not gonna miss too much, but we can certainly pop in and get more information on any of these topics. So as far as the kidney anatomy, internal, external, and function, anytime you name anything, you should know what it does, like name and function. That's kind of just an automatic. So with the medulla, you've got the pyramids, you've got the intermedullary columns. You should also know about the papilla, the bottoms of the pyramids, the relationship of the papilla to, say, the nephron or the collecting ducts, because the collecting ducts all converge on the papilla as far as the pyramid goes, and then that's what's dripping urine into the minor calyx and the major calyx. So you should know the calyxes, how urine, where the pathway of urine as it's formed by the nephron, where it's coming out through the kidney. The renal vessels, so we talked about the renal artery, and then you should know it goes to segmental and lobar, and then you have the interlobar artery, and then it turns over the arcuate where it's going above the pyramid, then the interlobular, which then turns into many different, branches off into many different afferent arterioles. Each of those afferent arterioles go into the glomerulus of the nephron, comes out of the glomerulus, goes around the, and the efferent arteriole going to the peritubular capillaries, going around all the tubules, getting all the reabsorption components, and then finally ending that area into the interlobular vein, arcuate vein, interlobar vein, and usually just a segmental, and then off to the renal vein. So that's kind of the nephron vessels in order. And you should be able to know kind of where they would be, either on a diagram or in a descriptive manner. The nephron anatomy and the function of each segment. So since this is just part one of renal anatomy, we're going to get into a little more detail on the nephron itself, but you want to consider what is the nephron, the big picture of the nephron, and where are these locations on the gross anatomy. So for instance, if we talk about the glomerulus and the Bowman's capsule, we know that's the renal corpuscle. Where in the kidney is that? That's going to be in the cortex. So it's sort of placing it in this big picture for the renal anatomy. Where's like the loop of Henle and the collecting ducts? They're going to be down in that renal pyramid portion. We should also know, kind of looks like here, I'm looking here, the renal anatomy, it's sort of looking more like my nephron list here, so I should have moved ahead. let me actually move ahead and see, because I'm seeing hormone action. So this is pretty specific to the nephron as we move along. So let me get out of this. Okay, there we go. And then move to the part 2 just to see. Where's the blood pressure hormones? So I must have, oh, I did put 1 and 2 together. Okay. All right, that makes sense. There's a nephron anatomy. Okay, so we'll go back up. All right. So
I have a quick question.
Yeah.
When we have to answer the questions, are we supposed to put like renal medulla, renal papilla in order to get it correct?
That's the proper term, because there's a papilla, there are papillas elsewhere in the body. Medulla, it could be like a lymph node has a medulla. So you always want to be clear, especially in that context. If you have the opportunity to be more precise and more specific, then definitely do that.
Okay, perfect. And then I'm sorry, one more thing. You said that you were going to, is this review with the with each of your slides somewhere on our Canvas website?
It should be. Is this right here, or like this list right here?
Well, yeah, like everything that you're going to be going through, is that just under like the slide notes just from the class, like the regular class?
Yeah, so like this list right here, let me just stop share really quick. And let me go to our website. Okay, and then I better look, because if it's not there, then I'll make sure it's there. So here we should be at our Canvas page. If I hit Unit 4, we should go here to, well, we could just do the PowerPoint notes for in class. Also should be the study guide too. So what I'm doing right now is this. Oops. Now it's doing something weird again. And is this slide right here. So I try to have the study guide and study checklist at the start of each section. So this one actually should be part 1 and 2, because the part 1 is, and then the rest of it's nephron anatomy. But so that should be here.
Perfect. I see that. Thank you so much.
Okay, and then, okay, I'm getting error messages on my Zoom. So if you're not seeing something, let me know. I saw something else on our Canvas page. I wanted to switch over. So let me just double check here where it says study guide. I'm pretty sure, I'm gonna say just open with that. And it should be this same thing. Yeah, so this is exactly the same thing. So we should actually just go, this would probably be easier just to go through the list here. So I have this study checklist. I put this in front of each of the sections. So since the kidney, I thought they were separated out, but they weren't. So I'll just stick with this particular list page. But
it's not showing anything.
Sorry? Oh, it's not showing anything?
No, except for Canvas.
Oh, thank you. So when I open up so then you actually didn't see the last one either that I just opened up. Okay, so I'll share that as part of the screen. I forgot, I didn't realize it stays with Canvas and then I'm opening up the new one. Okay, so you saw, actually let me go back to Canvas and really quick. So you saw where I went to the study guide. So I'm gonna wiggle the mouse across. So when I click that, I'm gonna stop share and then go back to, you guys probably already seen this here. This is the study guide, the study checklist.
Yeah, we see that.
Okay, so that's so that should be the same exact list as is embedded in the notes for the PowerPoint, which is exactly what the same list is that I'm going to go through today for your review. So, and then for the best way to study for this is if you go through each one of these items on the list and turn it into a question. Like literally, if you were to say, like if the checklist says, you should know about, so renal anatomy, if I go down to this 4th item down, filtration, reabsorption, secretion, action, and location. We'll turn it into a question and say, where does filtration occur? What is the function? You know, what is filtered? Where does reabsorption occur? What location does reabsorption take place? Or what components are reabsorbed? Every single one of these, turn them into a question. So they're more just topic specific, but if you turn them into a question, it'll be a lot more helpful for you to put that information into perspective. And then you can go through the notes and then sort of answer your own questions that way. And I would think that would be the most ideal way to study for this upcoming type of exam. So having said that, so we'll continue on here and we'll actually talk about filtration. So filtration does take place in it's going to be in the big picture in the cortex. That's because it's occurring in the glomerulus and Bowman's capsule, which is the renal corpuscle. So within there, so we're sort of covering filtration right here in the renal anatomy, but I will also be covering it as the first part of the nephron. I'm kind of blending them together. And so it's happening out in the cortex. It's right after that afferent arteriole brings blood into the glomerulus, which is really the capillary bed for this filtering unit. And then that Bowman's capsule is receiving all of the liquid it can pull out of the blood, all of the small solutes, things like glucose and various vitamins and amino acids, as well as a number of, a bunch of our waste. And so filtration is really removing all of these things. It's happening at the level of the renal corpuscle if we're dealing with the nephron down here. And if we're dealing with just basic anatomy, it's up in the cortex of the kidney. And then right beyond that, once we've removed all of this really good stuff, as well as a bunch of nasty stuff out of the blood into the tubules, the fluid is called filtrate. So now we've separated filtrate, that's within the tubules, and then versus the blood, which is in the vessel. So reabsorption takes place right away. We return the vast majority, over 95 to 99% of the water and other components, good stuff, are getting reabsorbed back into the blood. That happens in the proximal convoluted tubule, so that's on the nephron physiology side here. Our reabsorption location is going to be the proximal convoluted tubule. What we're bringing back are going to be like glucose, amino acids, vitamins. We pump back sodium so that water will also follow back. The processes would be through, there's a lot of ways this happens through diffusion. We have a lot of pumping action, facilitated diffusion. There's a lot of ways that we're actually getting things co-nutrient exchange. So there's quite a bit of mechanisms utilized that are in more of the basic cell process that is allowing for all of this good stuff to get back into the blood. And so again, this is taking place up in the cortex. The part of the nephron is the proximal convoluted tubule. And then we continue with our reabsorption into the loop of Henle. We go down into that loop of Henle. So if you're imagining filtrate now dropping down, it's leaving the cortex, it's going down into that renal medulla, and it's a straightaway down, and it's part of this hairpin turn coming back up. The first part of that tubule is going to be the thin, simple squamous, thin segment known as the descending limb, because it's going down into the medulla. That is where water is going to diffuse out. Water diffuses out more easily because on the back upward side, known as the ascending limb, and it's thicker because it has cuboidal cells, because the cuboidal cells are ideal for pumping, and they're pumping out sodium. So we have so much sodium trapped inside that hairpin turn that we can maximize water reabsorption. So we're still in the reabsorption story, but that's also known as counter current multiplication. And although we didn't go into the details of counter current multiplication, it is a way that we're actually maximizing and hyper concentrating the sodium so that we could maximize the removal of water out of the blood. So that's what that's referring to. And then before we move on to secretion, I'm going to touch up, since I'm jumping back and forth between this part 1 and part 2, the glomerular filtration rate. That's back on filtration. So on the filtration side, the rate of how effectively and how much volume the kidney is able to filter in a minute. That's what the glomerular filtration rate is. So it's really giving you a sense of the performance of the kidney. Where we have renal threshold, that goes to reabsorption. Renal threshold is how much do you have to have of something in our blood that we pulled out into the filtrate that we just have too much of, that you leave it left over in the filtrate. So what renal threshold would be is, what is the numeric number or the top value or amount of something, say glucose or a vitamin, I don't know, vitamin, K or something, that we have that is the maximum amount that we really can handle. If we go higher than this renal threshold number, we just let it stay in the filtrate and we pee it out. So the renal threshold is really the maximum amount of a given substance that we can actually retain back into or reabsorb back into our blood. Anything beyond that threshold level will remain in the filtrate and just leave the body via the urine. So the renal threshold really just is the maximum amount of something that we can have in our blood without peeing it out. You don't need to know the formula. You just should know just the value. It's just an average value of what would be a healthy GFR. So, so the renal threshold really gives us a sense of what we're bringing back in. And so if something that's good, say a bunch of amino acids that we spend a lot of energy eating and absorbing, and we have so much that we're actually peeing out a bunch of amino acids, we know we've exceeded the renal threshold for it. So good things that we normally would think are good for our body could actually end up in the urine. And that's just because we have so much of it that we just can let some of it go. Okay, so then we're moving along here, filtration, reabsorption, secretion. So we're going to do secretion here and also deal with the secretion down here. So secretion is stuff still in the blood, didn't get filtered, hanging out in those peritubular capillaries, even though stuff's returning to the blood via reabsorption. Secretion is, there are a few waste elements still in the blood that needs to get pumped down and get thrown out and put into the filtrate so our body can get rid of it. These substances tend to be larger and did not leave the blood at the point of the initial filtration in the glomerulus. So secretion is where we're more forcibly taking something from the blood and throwing it down into the distal convoluted tubule. In the sense of the gross anatomy, the distal convoluted tubule is up there in the renal cortex. So nephron physiology, secretion occurs at the distal convoluted tubule. That is, and the process, again, we're pumping something. Usually it's forcibly pumping. And you should know the type of solutes that are transported, like penicillin metabolites. There's a list that you should be familiar with that's in our PowerPoint slides that will tell you about the solutes that are transported, urea, a couple of other components. Let me see if I can find that list here for you. Here. Not too far, that's urine. Here we go. So the list is, let me switch over. Oops. Oh, stop sharing. I'm going to start to share this other list here. Okay, so creatinine, ammonium, urea, uric acid, drug metabolites, that would be something like I said, penicillin is an example, where it's breakdown components, we're sending out the urine, but they were too big to leave at the level of the glomerulus. So that's the secretion you should know. It goes from the peritubular capillaries to the tubules. It happens in other places, but we're really, the majority of it takes place in the distal convoluted tubule. Okay, let's go back to the checklist so I don't miss anything. So then once we're done with the whole filtration, reabsorption, secretion, and we did that where that's at, the solutes transported, what is finally leaving the filtrate, leaving the tubules, it goes into this collecting system, the collecting duct. The collecting duct is going to receive urine and it brings it all the way down from the cortex where the distal convoluted tubule ends, and it connects to the collecting duct in the renal cortex, but it's also that collecting duct continues down into the renal medulla. The end of the collecting duct is right there at that bottom little renal, the medullary papilla. And then after that, the urine drips out. So the collecting duct is really where it becomes urine. Now the hormone action that I have here in this list, in this upper one, as well as hormone action in this lower list, is to remind you of the hormones that are going to affect, and they primarily have an effect on the distal convoluted tubule. So there are 3 main hormones that you should know that target the distal convoluted tubule and have an effect on how much fluid makes it to the collecting system. So you would have aldosterone, an antidiuretic hormone. Although they come from 2 different places, you should be familiar with where they come from, what would cause them to be released, and then whatever their action is, and then therefore what would cause it to be shut down, like what is the body trying to achieve, and when it achieves that level, it doesn't need that hormone anymore. So that's the negative feedback cycle. So for instance, for aldosterone, it's going to get released from the body. We know that angiotensin II is one of the main ways aldosterone is released. It's released because blood volume is low. So the job of aldosterone is it comes out of the adrenal cortex. If you want to get real fancy, you can call it the zona glomerulosa, where the mineral corticoids are. And then aldosterone leaves there, goes to the distal convoluted tubule. At the distal convoluted tubule, it's reabsorbing sodium. Therefore, water comes in and we have a greater amount of blood volume. And then now, since blood volume was the goal, then it turns back and tells the adrenal cortex, we have our good blood volume. You can shut it off now. And that's what a negative feedback loop is. Antidiuretic hormone, its stimulus has more to do with solute concentration and dehydration. It's not so much worried about blood volume, although it's just worried about retaining water because you're dehydrated. And so when you retain water, it's just preventing you from losing so much water. So although they do exactly the same things, causes sodium reabsorption and therefore water reabsorption, they do it, their on-off switches or their negative feedback loops are different and they come from different places. And then we know atrial natriuretic peptide or atrial natriuretic factor from the right atrium is the only one that's actually going to dump extra sodium and therefore dump extra water into the urine. So let's go on and talk about urine. What is in urine? What is the composition and components? This part is really what I'd like to show you as far as the sediment and what's in urine. Let me see if I can find the slide on that I wanted to, that I think is helpful. Scroll down to the urine part. Not that one. Okay, this one's probably a better one. Okay, we'll stop sharing and back over to this. Cool. Okay, so this is the part where we're going to see things like red blood cells, epithelial cells. These are things that you would see, say, on a histological specimen, where we're actually pulling cells and stuff out. So these would be some of the sediment components of urine. So you should be familiar with that and know that these are normal things in urine. Maybe not bacteria, it's not uncommon, but it's probably not something normal or good that people would want in, or white blood cells, or red blood cells for that matter. You don't want blood in your urine, but you have mucus and crystals and casts and epithelial cells. That's totally normal and that you would find in urine, but you can find some of these other components in there as well. And then other things would be like glucose. You're never going to see it with the microscope. Ketones, these are more dissolved solutes. Glucose is only going to be in there if you've exceeded its renal threshold. Protein will be there if you've exceeded its renal threshold. Nitrates are different protein breakdowns. So these are things that you can test for in urine. And so that's what I mean when I'm asking about urine components. And then you should be familiar with this. The abnormal constituents, where you're dealing with what is the terms utilized. So you should know each of these terms and what they mean. Okay, so we'll go back to the checklist. Okay, we did the abnormal consent, and then urine transport anatomy and structures. So really, this is reminding me to tell you that you should know about the ureters coming down from the kidney. Already should know from the kidney anatomy, the calyces and the renal pelvis going to the ureters. The ureters are going to enter the posterior inferior portion of the bladder, just a little bit above the floor, but close to the floor. So you have two posterior ureter entries, and you have on the floor of the bladder, move a little more anteriorly, and then you have the urethral exit. So the floor of the bladder has this triangular shape that does not change. The triangular shape actually stays consistent even if the bladder is really full and expands like a balloon or recoils when it's empty. That trigone area actually stays pretty consistent. So it's through all the rest of that bladder that we see transitional epithelial tissue lining it, because that's going to help it expand or allow for it to expand as well as recoil, but still keeping a layer of epithelial tissues. And even in urine, I can't tell the difference, but people that look at urine regularly and look at cells, you can visually identify a epithelial cell and say, oh, that one's one of the transitional ones. Oh, this one's actually one that's up from the calyces. This, so you can actually identify epithelial cells that are sloughing off, because that's the nature of epithelial cells. They're built at the bottom like skin, and the top of our skin layer just flies off. Well, that's happening also along the urinary tract as well. So the structure, the most important structure of the bladder is that there is the trigone, as well as it's made of transitional epithelial tissue lining the bladder. Then you should know, going out the body, we have just the urethra. In females, there's one, the membranous urethra. In males, there's three. There's the prostatic urethra, then the membranous, then the spongy or penile urethra. Okay, so that's it for the urinary system. So I'm going to get off of that, and I'll move to the next slide set. And in fact, I'm going to let me switch it over, share screen so you can see where I'm going. I'm going back onto our Canvas page, and I'm going to click on study guide here on this next list, which is our next column. It says open, so let me switch over. Stop sharing, switch to the list. Okay, so here's our study checklist here. And again, the best way to study is turn each of these into a question. Components of intracellular and extracellular fluids, like you should just know those. So a question could be, describe the differences between intracellular and extracellular regions of the body. Describe the type of fluids that make up the extracellular fluid. That would be like cerebral spinal fluid or blood or lymphatic fluid, where intracellular is cytosol. It's just inside the cell. There are hormone effects and fluids on electrolytes. This is really the same exact hormones, the three main ones that you should know about: aldosterone, antidiuretic hormone, and atrial natriuretic peptide. It's really talking about how they move sodium, and then water will move. And it just depends on which direction. Are you moving it back into the body, or you're letting it go out? Electrolyte function, just what do we do with electrolytes in the body? Like really just big picture stuff on that one. What are some of the, why are electrolytes important? In class, sometimes we talk about the level of electrolytes in the body, and you can see dramatically the importance of electrolytes. There is, let me switch over. Electrolytes can actually stop your body from actually, or lack of electrolytes can stop your body from even functioning. There is a painful representation. It used to be, I don't really watch sports on TV as much more, but there was, used to be like ABC's Wide World of Sports all the time, and they had the agony, like the, you know, the wonderfulness of victory and the agony of defeat. And there was a particular female, Julie Moss happened to be her name, that was finishing the Hawaii Ironman. What was this? Probably in the 80s, early 80s perhaps. And she was an amazing triathlete. Obviously, she's an Ironman, she's winning, and she had won. And so in this particular one, she had a competitor that was racing ahead of her, and she was not, in trying to keep up and not let this person get too much of a lead on her, she was not drinking enough and replacing her electrolytes. And so the sort of agony of defeat video clip that's sort of seared in my mind shows her literally crawling down Ali'i Drive, which is the last, you know, a couple miles before you get to the finish line for the Hawaii Ironman. And she's crawling hands and knees. Her legs are complete noodles or complete spaghetti. She can't even hold that. And that's because she sweated off all of her electrolytes. So that's a super dramatic representation of how they're important. So in our slides, you should just understand some of the functions: muscle contraction, nerve action. So she can't even control her muscles, and she probably didn't even have enough ATP to balance out the calcium inside the muscles. So again, there's a list of just generally what electrolytes do. Specifically, I'd like you to know about sodium and potassium in a little more detail, so that way we're not diving down into too many. And the most obvious things for sodium and potassium we know is going to be for nerve action. So sodium's involved in depolarization, potassium is going to be involved in repolarization. So if you have an improper balance of either one of these, you won't have nerve function. And if you don't have nerve function, you're not controlling your body, or your heart's not beating. And in fact, solutions that involve euthanization are usually sodium channel blockers. Anyways, so your very life essence is critical based on these electrolytes. So then the other part that's probably more important of this chapter is the acid-base balance. You should know this is just more what acid is, to be able to describe an acid. You should know, where does acid come from in our body? What are the sources of acid? So we talked about ketone metabolism, we talked about lactic acid, we talked about protein metabolism, fat metabolism, that is again the formation of ketones. So and then carbon dioxide, just making carbon dioxide actually drops your pH a little bit. So these are some of the sources of acid. Excuse me. But then what does our body do about it? We should know the buffering systems. Here's this last one, the buffering systems and regulatory mechanisms. So breathing, we can breathe it off. We also have large molecules in the blood that we bind the little H pluses or the protons to. And then if we want long-term mechanisms, we really want the kidney. The kidney says, hey, we've pulled this filtrate out, and there's a bunch of H pluses around here. Let me bind them up and not let it get returned to the blood, and so I can take it out into the pee. So you should be familiar with the buffering mechanisms. That's the most important part of this particular chapter. Okey-doke. So now we'll go on so I'll click on share screen, we're going back over to our Canvas page here, study guide, open with. Okay, switch over, stop share. Okay, so here we're going to deal with the female-male reproductive system. So pretty straightforward. There's just a lot of details in there. You know, for both of them, we want to know, how do we make the gamete? If male, the gamete's going to be sperm. Female, it's going to be an egg. So big picture stuff. How do we even make that? How do we make it? And how do we deliver it so that ultimately the egg and the sperm get together? What are the parts that we have to go through? So that's kind of our big picture concept here. And again, with this little checklist that we have, I went through these are the areas that in the lectures, whether it's our regular exam and I have a multiple choice questions, or I'm going to ask about an open-ended question, these are still the topics I find most important from your lectures that I would have you focus on. So again, the best way to study this is just each of these checklists. Uterine tube anatomy, something pretty as innocuous as that, turn it into a question. Name the parts of the uterine tube or fallopian tube. You know, what's the function of the fallopian tube? What's unique about the fallopian tube? Those are some of the questions that could come up. So again, just wanted to put it in perspective. The more you can sort of turn these into questions, it opens your mind up to different ways that you could answer or look at this topic and better understand it. So let's get started with, so we're going to go down and we'll start with the male first, because I always find it easier to end with the female so that we can move on to development. And so it sort of blends that way. So we're going to start down below, hit the male anatomy. Okay, that's pretty wide open. Just know about male anatomy. So, you know, this is sort of big picture stuff like, okay, you have testes, you have the scrotal sac, you got some, you know, muscles that are going to hold it closer to the body for either, you know, warmth or for ejaculation. So we've got muscles involved. You have the tube that the sperm's going to make it back into the body. You know where the ampulla is. That's going to be on the backside of the bladder. Then next to it, you're going to have seminal vesicles. So when you think of seminal vesicles, you think, oh, that's just the first of 3 of the major glands for males. So you've got seminal vesicles next to the ampulla, sitting there on the back of the bladder. And then at the point of ejaculation, the fluid from the seminal vesicles go down into the ejaculatory duct inside the prostate. At the same time, the sperm is coming from the ampulla also into the ejaculatory duct. So you're mixing seminal fluid and sperm together in the ejaculatory duct, and the prostate itself is also adding its components. So right away, you're getting quite a powerful mixture. The mixture that's involved in that is going to then target the sperm. And actually, when we talk about sperm anatomy, we're going to talk about the midpiece on the sperm. That's the motor, and that's actually going to cause the tail to move. So the sperm becomes activated only once it's starting to mix with these glandular substances, which happens at the point and the moment of ejaculation. So as we go through the whole male anatomy, we're sort of jumping around these topics because you want to sort of follow the process. So if you want to start with either making a sperm, okay, in the testes, seminiferous tubules, you're going to need to know the cells from the wall inward in terms of developing a sperm, as well as the chromosomes involved, the number of chromosomes each cell is going to have, and then where the sperm is going to go. It makes it through the seminiferous tubules, heads up to the epididymis, wiggles through there. That's the place of maturation. The sperm starts to develop a little bit more. Think of the testes of where sperm are born, and then the epididymis is where they're sort of maturing and developing. They're still it's on a conveyor belt. They're still not moving themselves. We don't want them to move a lot. And then up the vas deferens, and then hangs out at the end of the vas deferens on the back of the bladder in the ampulla, and it sits there and rests until it's asked to leave the body. And if it never leaves the body, it will eventually be reabsorbed by the body and doesn't go anywhere. So you can follow sperm production that way. Then you can, go from the point of saying, all right, we have sperm hanging out there in the ampulla. It's going to get ejaculated out of the body. What's going to happen in that situation? So that's where you're thinking sperm in the ampulla, again with the seminal vesicles. Next to it, ejaculation, the fluids from both seminal vesicles as well as the sperm in both ampulla go down into the prostate through the ejaculatory duct. Prostate's adding its components to the seminal fluid, goes down through the prostatic urethra, through the membranous urethra. Right outside the membranous urethra is the third gland, The bulb urethral glands, well, they had already sent their substance out because that bulb urethral or Cowper's glands already sent a bunch of solution through the penile urethra to neutralize any remaining urine in there, to reduce any acid, and to create lubrication, early lubrication. So all of that's coming into play, and here comes some sperm mixed with seminal vesicle fluid, mixed with prostatic fluid, barreling down and out the penile urethra. So that is that process. Then we have, let's just basic penis anatomy. Well, in order for an erection to take place, you have to have the two dorsal strands, the corpora cavernosa, on the dorsal surface of the penis. Obviously, blood's going to come in, and during arousal, you're going to have increased blood flow in, but we also restrict venous flow out. So it's really like blowing up a balloon. You're just not allowing any of that pressure to leave. So it's more about venous constriction in most cases, as far as creation of an erection, and then fluid escapes out into that corpora cavernosa space, and that's what actually causes it to increase in size and hardness. But then you have corpora spongiosum. It's also erectile tissue, but it's not meant so much to inflate and create hardness as much as it is to circle and surround and protect the penile urethra, because you don't want that to get constricted off. You don't want to obstruct that. So corpora spongiosum is going to actually protect that so that the semen can actually make its way out. So then, bottom list of the male, we've got hormonal regulation of sperm cells and testosterone production. This is just understanding the role of follicle stimulating hormone from the anterior pituitary and luteinizing hormone. Follicle stimulating hormone is going to stimulate the spermatogonia in the seminiferous tubules to form sperm. Luteinizing hormone from the anterior pituitary is going to target interstitial cells, also known as Leydig cells, to increase testosterone production. Okay, there you go. There's male anatomy in a nutshell. Did we miss anything? Nope. Okay. Any questions? All right. Yep, go ahead. Thought I heard some noise. Okay, please interrupt me if you have questions. Because if I just barrel along. So off to the female anatomy. So we've got, again, you want to put these things in question form. There's ovary anatomy, the ligaments, functions. So the point of, let's see, the function of the ovary, that's kind of a weird thing to say. So as far as you should know, the ovary sits out on its own. It's tethered to the uterus by the ovarian ligament. So it's kind of like hovering out there. But as far as the ligaments go, I'm going to pop up a picture. Those ones, you might need to visualize that. Let me find, scrolling down in the slide set to find the ovary, or just the female reproductive system. Okay, that's a better picture. Okay, stop sharing, go there. Okay, so in this picture, here we have our ovary, and this guy here is our ovarian ligament. So it's from this medial aspect of the ovary towards the uterus, and on this side we see it from the medial aspect towards the uterus of the ovarian ligament. We have outward from the ovary out lateral to the wall, and you can see it on both sides. It's the ligament as well as blood vessels, and that one is the suspensory ligament. And then we have, you don't see it in this picture, a uterosacral ligament that's actually going from the uterus, because it tells you exactly what it does, and goes back posteriorly to the sacrum. So it's actually tethering it to the back of the body. And then this guy, the sticking out right here, coming forward, that would be the round ligament. So it's coming more anteriorly. So those are some of the ligaments that hold it in place. And one of the unique things, the ovary doesn't have specific features. We're going to spend some time talking about the follicles developing inside the ovary. But it's not connected to the fallopian tube. So this picture on the portion on the right side looks like it's just hanging on top of the fallopian tube. But on the left side, you can see that the fallopian tube, or sorry, hanging on top of the ovary. On the left side, you can see that the ovary is detached, is separate. So when the egg is actually launched out of the ovary, it has to cross a space. It sort of has this very much a leap of faith to then enter the fallopian tube itself. So while we're there, I know that's on one of our lists to know. So this fallopian tube, also known as the uterine tube, you can call it either one, means the same exact thing. Traditionally, it's known as the fallopian tube, and it's the more common term, although the more proper sterilized anatomy, term is more just uterine tube. I think that anatomy is trying to drop the terminology that recognizes the anatomists that discovered them or named them. So they're sort of removing that from, I find a little bit sad because I quite enjoy history and some of those elements, but you can use, when you write your answers, either one of those terms. So we have the fimbriae, that's the finger-like projections at the very end. The fimbriae actually move and undulate so that when an egg is launched out of the ovary, the fimbriae are going to try to gather it in. And so then it brings it into this isthmus, just sort of this first wide part, and then the ampulla. And the ampulla is right along here. That is the primary site where fertilization takes place. So if the female has sex a couple days ahead of time from when she ovulates, by the time the egg launches out and gets right over here, it's within the first 24 hours, if sperm is already in that location, that's the most ideal time to get fertilized. Now, if you're trying not to have an egg fertilized, then don't think that everything's a-okay until you ovulate. The closer you want to be a little cautious, the at least 3 up to 5 days ahead of ovulation. So that doesn't give you a lot of time. So you have so it's in the second week. So again, depends on your perspective, trying to get fertilized egg or trying not to. So, so we have the ampulla, and then it brings us into the rest, to the uterus itself. Let me pop back over, stop share. Pop back on over to our checklist.
I have a quick question.
Yes.
The broad ligament, is that sheet that comes out?
Yeah, good question. Thank you. That's what a very important ligament. I was sort of looking at all the other ones. I meant to end with the broad ligament. Yes, the broad ligament literally is like laminates all of that together. And the pictures don't do it justice. And in class, in our lab, I have a uterus in a bucket that I usually pull out. And it's fascinating because it's where the broad ligament's been cut. If I hold both sides and I just hold it out, you can actually see light through it. It's a pretty thin membrane, kind of like the pericardium, if you guys recall that. I don't remember if we showed that in our class earlier, but it's thin and it's really tough. But when you hold the broad ligament out, you can actually see the suspensory ligaments. You see the fallopian tube, the ovaries even embedded in there. They're sort of just in place. And so, like I said, it's sort of laminated in place. So yes, thank you for bringing that up. That is an important ligament as well. Then meiosis and the corresponding follicular phases. So you have to know for the female, and you should know meiosis, so down here too, but male meiosis is easy. Female meiosis, it just takes forever to get this thing done. So it is in a halted state of meiosis. So in the female reproductive tract, it's not developing, it's not going to be completed. It's actually stops meiosis 1. Meiosis goes through 2 levels. There is, let me switch over here to this. So you have meiosis 1 is only from the embryo to puberty. So literally the embryo when you were in your mom's belly, if you're a female, all the way into puberty. It's like these eggs, on the right, I have it in red here, where it just stopped. And so it allows for that egg to not deteriorate. as quickly. So it's sort of like, this is a really stupid analogy, but it's like, you're not going to keep a bunch of cupcakes, for 50 years. So maybe you'll just let it sit with the flour and the, sugar stage. It's not quite fully made, but you're a lot more likely to prevent degradation. So in the female, because she's given all of her gametes at the point that she's an early embryo, unlike males, they just churn it out from the spermatogonia. And so there is no sort of degradation over time. So it's important to know that it stops in meiosis 1. After puberty, the specific eggs that are brought forth to then be, sort of a candidate for ovulation, they then mature up to the point of metaphase 2 if they then ultimately get ovulated. And so again, it's still in the blue square off to the right. So it's still not done, actually. And it's only at the point that once a sperm penetrates the zona pellucida, that's the protective layer around the egg, once it's been released and it's in the ampulla of the fallopian tube, that point, when it penetrates, all of a sudden the egg finishes meiosis and then quickly gets the chromosomes lined up so that the male chromosomes can line up and then can progress from that point. So that's what's important there. Then I'm switching back over. So that was my, and then we'll do the corresponding follicular phases. So the follicular phases, this is just the follicular phase. During the follicular phase, you go from your primordial to primary. So let me see if I can find the picture for that. So you should know these names: primordial follicle, primary follicle, secondary follicle, and mature. So we'll look at it in this picture where we know the primordials are located in the periphery of the ovary. They become, its cells enlarge immediately around the egg, but it's still tight against the egg, so it's a primary follicle. So this is happening in the very first week of development. It so happens during this first week of development and the first week of the cycle for a female, that's the same week that in the uterus, menstruation's taking place. It's getting rid of last cycle's endometrium. So the removal of last cycle's endometrium, menstruation, corresponds with the new cycle's new batch of eggs coming into the primary follicle stage. Then as we go into week 2, ending week 1, going into week 2, we start to see an antrum form. That's a little fluid-filled capsule starting to form. It gets larger and larger and larger. And then as it gets bigger, this secondary follicle, it starts to move towards the edge of the ovary. And it gets even bigger than what we see in this particular image. And it looks just like this secondary follicle, just a little bit bigger, and it comes right to the edge. And that's the point of a Graafian or mature follicle. And we've got the egg, which is the light pink inside, surrounded by the dark pink zona pellucida. And then some of these black looking cells, then they're sort of, the edge is docked here, but we're just talking about the black looking cells immediately around the dark pink circle. That is the corona radiata. And that is what actually leaves the ovary, takes that leap of faith. The fimbriae of the fallopian tubes gather it up, and then we have what's left over. And so this large outer group of black clustered cells are known as the follicular cells because they make the follicle, that's the bubble. And so once this inner egg leaves with its corona radiata and the zona pellucida, and we have only the follicular cells here left, they puff up and become the corpus luteum. And then we're now in the luteal stage. So the follicular stages, well, it's the follicular stage. So this should be singular. In the follicular stage, we have, these are the cell types, or stages of the follicle might be a better way to say that. So this is occurring during the follicular phase or stage. Then the corpus luteum where is that? Well, we can see kind of that in this little picture. So once the egg leaves, this remaining follicular cells puff up and become the corpus luteum, and it gets so huge. In this case, it almost takes up the whole entire ovary. So it becomes this giant secreting gland that makes progesterone. Okay, I'm clicking back over to have a little look here at,
I have a quick
yes.
question. So it puffs up like that after the egg is ejected, correct?
Yes.
So it starts off way smaller than that. And then as soon as the egg is ejected and that the hormones start producing the progesterone or whatever, then it starts puffing up like that and filling up that space.
Yes. So when the egg is, here we go, this one, it's these cells around out here. So there's, see how they're packed in this wall. It's kind of thick. So not so much this side. You can see here how tiny the egg is compared to the last slide that we looked at. So this is a mature follicle, how they've moved over to the edge here. Maybe I'll pop it up so I can draw on this. So this part gets ovulated. And then let me change colors to green. And so these cells that I'm sort of scribbling on in green, these remaining cells are known as follicular cells. They then enlarge and become the corpus luteum, which is that. So this is where they're huge. It used to be just like that big when I drew it the first time. And then now it enlarges and puffs up. And that's
almost like taking up the whole ovary, right?
Yeah, it's huge.
Okay.
Like
Okay.
here's an actual ovary. In this case, it's only taking up like a third of it perhaps, but that's
Wow.
corpus luteum. This one is really big, and I suspect, perhaps, it doesn't say when I got it, this is an old slide. I actually suspect this would have been more from a pregnant female where it had been secreting for a really long time and maintained, because it is on the bigger side, but it does take up. So this one's more of this female probably either had her ovary taken out for any reason or died, and they had got found at postmortem, but she was definitely in her luteal phase because she has a very prominent corpus luteum. And in this one, notice that is actually last month's corpus luteum that hasn't quite, it's not sending out progesterone, but it's still so big, it actually takes a little while for it to completely disappear.
Okay, thank you.
You're welcome. So let's go over to our checklist here. Okay, so we've got the uterus. So the anatomy of the layers, we'll do layers. The serosa is the connective tissue on the outside. Myometrium, that's the muscle, it's the thickest part. So that's the main part of the uterus as far as musculature. When you're looking at it in a gross anatomy, more visual way, you're like, cut it open, like the heart. You know, we have that tiny layer of the endometrium, sorry, not the heart, the endocardium and the epicardium, but the thickest part of the heart is the meat, right? And so that's myocardium. Well, here we have the myometrium. So that's going to be the main layer. But the innermost layer, the endometrium, is actually the most important layer, but it's a small, thin layer, relatively speaking, but it's the endometrium that has the most action, if you will, because it's developing. You know, we have the early, during the proliferative stage, it's building up. Then during the secretory phase, the progesterone makes it start to ooze out nutrients and the glands become really active. And then nothing happens, no implantation takes place, and then it has the menstrual phase and gets rid of it. And then it starts over again with the proliferative phase, build up a new endometrium. So endometriums are a very dynamic place. Every week something's happening that's different, except for the luteal phase. It's not anything different because that's 2 weeks of the same thing. So those are the layers there, and we'll talk more about the endometrium specifically. We already talked about the ligaments, the uterus anatomy. The top part is the fundus. So when a female is pregnant, it's literally that top part that you could palpate actually. Once it gets big enough and it rises up beyond her pelvic basin, then you can palpate the top of the uterus and you're touching the fundus of the uterus. And then the bottom of the uterus is the cervix. So the cervix is really the entryway into the uterus, and the cervix is located in the vagina. So that's when you're getting a pap smear. It's just a long Q-tip, swipe the cervix. And so it's really part of this dynamic uterus, but we have access to it. And the reason why it's really important for females to get a pap smear every year is because it's so many changes can happen that it's also likely that changes could happen wrong or incorrectly. So it's a hotspot for cancer. As well as the endometrium could go wrong. So it's really important to have those evaluated, and it's very fortunate that you actually have the cervix that's so accessible, if you will. Maybe not ideally accessible, but it's still accessible nonetheless. Okay, so those are the parts of the uterus. External genitalia, really don't do too much with that, but I just want people to know their way around there. So, you know, you've got the clitoris, which is the most anterior portion that's made of erectile tissue. The clitoris is got erectile tissue that's similar to the male penis. And then just back from that would be the urethral opening for the bladder, for the urine. Back from that, you have the vestibule, which is opening space. And that goes, then enters into the vagina. There's a bunch of lubricating glands within there. I'm trying to think. And then the labia, those are the skin folds outside of it. So you have a smaller, minor labia minora, and then you have the larger outer folds, the labia majora, which also have erectile tissue within it as well. So we use we have the same parts, male and female. We just sort of configure them in different ways. Mammary glands, again, we talked about the 4 phases of mammary breast development, where, you know, boys and girls are at stage 1. Boys and girls have the same amount of glandular tissue. It's only girls, because they have a large estrogen surge at puberty, starts to initiate early development of this breast tissue. And only after they get pregnant does then hormones from the baby, from the placenta specifically, go and target the breast tissue and say, you need to go into stage 3. We're going to have to get ready to lactate and actually make milk. And then finally, when the baby gets born, then it moves into stage 4, where then you're actually producing milk. And then once the breast tissue has gone through all those stages, it is actually in a more protective nature than somebody that hadn't had a completed pregnancy, or someone, so one of the more higher risk folks for breast cancer would be that they had gotten pregnant and had yet to have a complete pregnancy. So whether, you know, it was an abortion or a miscarriage, they hadn't had another pregnancy. So that would be really, the breast tissue is like, I'm kind of in the middle of growing and developing, and now I'm stopped. And so that growth can then go in a more volatile way. So until that female has a completed gestational cycle and the baby's born, then would finally enter stage 4. And beyond that, whether you have 1 kid or 10 kids, that doesn't really matter beyond that. So that's, and the hormonal stimuli, you should know from Unit 1, prolactin tells the breast tissue, make milk. That means promoting lactation. And then oxytocin tells the ducts, let's send the milk to the nipple. And so that's the story with those 2 hormones. While we're talking about hormones, we'll go to follicle stimulating hormone and luteinizing hormone. Follicle stimulating hormone from the anterior pituitary gland is going to go tell those primordial follicles, let's time to go. We're going to pick a few of you guys as candidates for ovulation. So that's follicle stimulating hormones initiating that process. It's initiating follicle development. Luteinizing hormone is helping to cause estrogen secretion. There is a different or secondary luteinizing hormone surge mid-cycle that corresponds and facilitates with ovulation. But I really don't focus on that as much, because I want to try to make it as even to males and females as possible in your thought of just knowing follicle stimulating hormones gonna make the gamete, whether it's an egg or sperm. luteinizing hormone is gonna make the sex hormone, testosterone or estrogen. So at least you have, one's a hormone maker, one's a gamete maker. So kind of just that big picture view there. And the biggie, so these last two parts are the big deal, maybe not a big deal, but the more confusing parts, I think, for the female reproductive system. So we're going to talk about the uterine and ovarian phases. So let me stop sharing here and see if I could show you something a little more interesting. We'll go to this here. We'll share that. Okay. So on here, we can see these are the endometrium phases. So in the bottom, let me pop this up into a slide thing. I'll draw on it. Oops. No. Let me do it. There we go. So we have, obviously from this is day 0, day 28. So this is a timeline. It's sort of just drawn weird. It looks like, so it's sort of each day is in a calendar. So if we do 0, 1, 2, 3, 4, and so on. But so this here is an excerpt from that portion where it's saying this is the menstrual phase. We've removed the endometrium, that's this part here, gotten rid of it, and we're right down to your myometrium. So the endometrial layer is pretty thin and just really just gone away. It's been destroyed. Then we go into sort of this rebuilding, phase here, and that's going on this part. So what we see are glands that are just little round. They might get long and turn into ovals, but they're really angular and more geometric looking, because this is the proliferative phase. During the proliferative phase, it's dominated by estrogen, and estrogen is causing the building of this endometrium. And so there's other hormones involved, but estrogens are primary growth promoter. Then we finally get at the moment of, and this at this point here, we'll sort of draw a little line in our timeline, and I'm going to type in ovulation. So ovulation takes place, and so after this green line, we have progesterone present. So I'm just going to write progesterone. I might as well write estrogen on the side. Okay, so the progesterone present is actually telling these little glands here start to fill up and fill these little spaces, and I'm drawing them with green, with this glycogen nutrient stuff. Like we've got like just tons of little granola bars we're making for an implanted egg potentially in there. So all of these are food sources that a potential fertilized egg, and specifically the trophoblast portion of it, because if an egg gets fertilized and it becomes a blastocyst, it will actually embed into the wall. This would be where the little egg is, and it's a blastocyst. So remember, it's got the embryoblast and the round trophoblast. And then what happens is that trophoblast, instead of a nice round outside, it sort of makes these finger-like projections because it's actually tapping into, these glandulars. So it's feeling around. It turns these little fingers and it's kind of embedding and invading the endometrial wall, while the embryoblast that's right there is starting to develop into its germ layer thing. So we've got progesterone, and so this secretory phase of the endometrium is obviously made by the progesterone, and we see it because it looks a lot more squiggly, and so that tells you that you have an active producing gland at that point. So you should be familiar with, maybe while I'm on this slide, even though it's going to make it hideously messy, what color? Maybe I'll use purple. So if we have our menstrual and proliferative, this section here, during our menstrual and our proliferative in the uterus, is the same as our follicular in the ovary. Then the secretory, which is the last 2 weeks, I'm going to put a little square here, just going to write over. This would be our luteal phase. So I really made a mess of this slide, but you can see perhaps a little bit better the way the slide goes. So if I was going to draw a picture of this, and if I were going to take this and make it into a study guide, I may or may not, if you find this middle picture helpful, maybe have that little middle picture, make sure you have the days on it. And then on the bottom part, maybe include these same pictures. And then on the top part, you can include a picture of the ovary. Under the follicular part, I'd put a picture of the ovary showing the primordial, probably like that one, that cat one with the purple, where you can actually see the primordial, primaries, and secondaries. And then on this side over here that I'm scribbling on, I'd put the picture of the corpus luteum. And then you can see it's the corpus luteum and it's making progesterone. And then you really have all of it in one place. Okay, so I'm gonna stop share. It's now looking at it, looks like you can get a headache looking at this thing. Okay, someone's got a question. Yes, and so you should know with the pregnancy hormones, when we get to development, that there is the inhibin and relaxin. Those are ones that were also coming from the placenta that will facilitate the development early and early labor portion. So that should be on your list too. Okay, so let's see. Back to our checklist. Make sure, I think we're about done. Yeah, so we kind of went through all of these, the predominant hormones. I didn't mention, as far as the predominant hormones go, the menstrual phase. This really just has a low, isn't known by a predominant hormone because it's low hormone. It's really like nothing. And then as the days go on, we get to day 2, 3, 4, estrogen starts to, rise. And so estrogen starts to develop, even though we're getting rid of the old endometrium, we're starting underneath to rebuild and get the new endometrium formed underneath. And so then you have like low levels to increasing estrogen. Then you have the proliferative phase, is marked with high estrogen levels. Then you get into the secretory phase. You still have estrogen there, but in the secretory, it's eclipsed by the great amount of progesterone that's there. Okay, so that was all together. So we'll stop that part, and then we'll get into the very last portions. Let me click open this, the last study guide. Okay, open here. Okay, let me switch over, do a screen share with you guys. A lot of clicking here. Okay, so in this last one, we've got the cleavage sequence. So I don't think I used that term the other day. Cleavage is the terminology used when the cells are dividing. We talked about from the point of a zygote, and then it becomes a morula. It's developing, it gets to the morula stage, and it continues developing, and right before it becomes a blastocyst. But it's increasing cell number exponentially, but this overall size, it's not getting bigger. It stays within the same egg size, and that's known as cleavage. So it's the divisions, but again, stay at the same size. So then we can want to think of the timing. So at day 0 is fertilization. And then you have about, we'll just say roughly, it could be 5 days, but roughly a week. We've gone through the morula stage that we ultimately get to the blastocyst stage. So then we have the blastocyst stage, and that's the point that you have to be at for implantation to start. So once implantation starts, then you should, also for the blastocyst, it's critical that you know the 2 parts of the blastocyst, the trophoblast and the embryoblast. So then okay, the trophoblast, it's going to release a hormone. That hormone is known as human chorionic gonadotropin. HCG targets the ovary. The ovary continues to make progesterone, and it goes right past the 2-week timer that would normally be in place for the corpus luteum, and it allows it to continue for at least several months, at least about 3 months, well, it lasts about 6 months, but with decent secretion of progesterone for the first 3 months for sure. So then So that's the hormone release. The inside of the embryoblast is going to then form the germ layers. So you should know the ectoderm, mesoderm, and endoderm, what organs come from each of those 3 layers. Then you should know, as this forms, curls down, forms the tube, we start to develop also extra embryonic layers. We've got the allantosis, we have the yolk sac, we have the amniotic sac, chorion, placenta. You should know about those, where they're placed. You would see the placenta structure and hormones. So the placenta structure is really chorions on one side, placenta with the villi, and it's kind of looks like a wet, if you saw placenta after it's out, it's sort of one side's matted like wet dog hair, it's gross, and the other side is just smooth like a membrane. The smooth side's the baby side. The wet, nasty, glommy side is what actually was interfacing in the uterus on the endometrium side to the mom. Then you have the umbilical, 2 umbilical arteries leaving the baby, going to the placenta, and a single umbilical vein coming back in. When we get to the first trimester, you should know the most important one would be the neural tube. That's 3 weeks. And what do you need to make sure that that gets sealed up? You need folic acid. So that's important. I want you to know 3 weeks at 3 weeks, that actually corresponds exactly with the menstrual period, or what would have been the menstrual period for that female if she wasn't pregnant. So for a female that is pregnant, when she's sitting around going, I'm missing my period. Maybe I thought I was on my period this week. There's a neural tube being made. Somebody's brain and spinal cord is being formed at that point. And that's the point where it's really critical that you have the folic acid in place. Heart cells, they start to develop. So you should know these would be the good benchmarks. So I don't want you to memorize everything in terms of development, but as we go through, you should know, the skeleton is fully ossified by 16 weeks. You should know that by 12 weeks that the external genitalia is formed. So therefore, if there's a penis, you could see it. So that's usually how they sex the child. It's either presence or absence of a penis. You should know about the limbs with the little hands, where they're like little mitten hands, and then they got little ridges, and they got cartoon fingers, and then finally they look like they have real fingers. So you should have that idea. And then you should know the difference between twins, monozygotic and dizygotic. The dizygotic are known as fraternal twins. The monozygotic are known as identical twins. And you should know, they come from, the identicals come from 1 egg and 1 sperm, where the dizygotic, that's how it says, di meaning 2, zygotes. So it has 2 different eggs, 2 different sperm. They just they happen to just hang out in the uterus. They're the same as any other sibling. They just happen to be occupying the uterus at the same time. Okay. We kind of rolled through that. It was a lot of information. So hopefully you guys feel comfortable enough with the information that as I was rolling through it, although it was pretty quick, it was a lot of material to go through. So I didn't need to be too fast. I just wanted to make sure we hit the highlights. And it's my hope that most of you guys are sitting home going, yep, I got that. Yep, I got that. It's in my notes, or jotted yourself down some notes of some areas that you might want to look at a little bit more closely. And that's it. So any questions?